I'd like to welcome everyone to the Charles F.A. Culling Memorial Lecture. For those who don't know who Charles Culling is, Charles Culling was a man of limitless energy, endurance, wit, and compassion, who always found time for anyone who needed advice and direction. He was dedicated to the field of laboratory science, starting in the Royal Army Medical Corps as an officer in charge of training of medical laboratory technologists in India during World War II. He pursued his love for histotechnology as an appointed professor at the University of British Columbia. When Charles lectured the students, they were entertained with his witty stories and his famous collection of amusing slides. He advocated the principle that a lecturer's objective was to turn the student on, and thereby creating a desire to learn. Perhaps the most important aspect of his lecture was he never failed to see the humorous side of life. Charles continued his passion to write the written word as illustrated by more than 80 publications. His contributions to the field of histotechnology, especially histochemical techniques pertaining to tumors, are globally recognized through his textbooks titled Handbook of Histopathologic Techniques. He was particularly proud that all of his published procedures were reproducible from his personal experiences. Like all great inventors, he believed that an important experiment is one that gives the unexpected result, and he was on his 4th edition when he passed. He was Dr. Culling in all scientific circles he was involved in, even though he never formally achieved the final degree, and his death was a great loss to all who knew him. He was a kind man, and his contributions, both personal and scientific, have had a lasting impact. In lasting memory, in his contributions to the National Society of Histotechnology and in the field of histotechnology, the Charles Culling Lecture was established and has been a memorable keynote lecture series since it was presented in 1982 in the symposium in Boston. This year's beneficiary is a woman also of limitless energy and endurance, but moreover a visionary, much like Charles Culling. She definitely is not afraid to challenge the unknown territories of cutting-edge science. It is a great honor to introduce this year's recipient of the Charles Culling Memorial Lecture, Dr. Anna Barker. Dr. Barker served as Deputy Director of the National Cancer Institute, NCI, and as Deputy Director for Strategic Scientific Initiatives for 8 years, where she developed and led or co-led a number of transdisciplinary programs, including Nanotechnology Alliance for Cancer, the Cancer Genome Atlas, Clinical Proteomics Technology Initiative for Cancer, and the Physical Sciences Oncology Centers. Dr. Barker was the founding co-chair of the NCI FDA Interagency Task Force, founding co-chair of the Cancer Steering Committee of the Foundation for the National Institute of Health Biomarker Consortium. She served as a senior scientist and subsequently as a senior executive as Battelle Memorial Institute for 18 years, and co-founded and served as the CEO of a public biotechnology drug development company. Her research interests include complex systems, biomarker discovery and development, experimental therapeutics, and free radical biochemistry in cancer etiology and treatment. Dr. Barker completed her MA and PhD at The Ohio State University, where she trained in immunology and microbiology. She currently is co-director of Complex Adaptive Systems Initiatives, director of National Biomarker Development Alliance, known as NBDA, and is a professor at the School of Life Sciences at Arizona State University. It is a great honor to present Anna Barker for this year's Culling Lecture. Dr. Barker.
Everybody turn their phone off for sure. Good morning, and thank you very much, Beth, for that introduction, and thanks to the organizers for this privilege. When you give a keynote lecture like this, it's a little daunting because I didn't know Dr. Culling. I'm much too young to have known him, obviously. I did my research, and I was impressed to find that not only did he invent and actually, I think, envision so many things of what we still use in histology, what you use and what we use in pathology, but more than that, he was a visionary, and he positioned all of us and all of you for the future. The ironic thing is my area of specialty is cancer research, and I've spent my life there. And the irony to me was that Dr. Culling died of lung cancer. And that was most unfortunate, but unfortunate though it is, I think, the plague of the 21st century. So I'm going to today just take you through a few things, and I'm here really for a reason. This group holds the key to personalized medicine. And I'm going to convince you before you leave today, otherwise I'll lock the door and no one will be able to leave, that it is this society, it is this group that holds the key to us successfully turning all that we've learned and all that we have supported for these many, many years in biomedical research into real advantage for patients. And by real advantage, we talk about cures for diseases like cancer. Those are still very much in the future, unless we do some things quite differently and unless we change the way we do things. And this is, I'm hopeful, an organization that can help us to do that. So I'm going to tell you a little bit about cancer, which everybody should know more about because it's going to be the disease that takes out most of the people sitting in this room. I'm going to talk a little bit about what personalized medicine is and the critical nature of biomarkers in this whole space. Those two are unbelievably linked, and you can't have a personalized or targeted therapy without a biomarker. A biomarker essentially is critical, and therein lies the problem. And I want to tell you a little bit about the state of the science. I had a lot to do with helping us to get to this stage, especially in terms of cancer, but also in biomedicine overall. And then I want to tell you a little bit about this National Biomarker Development Alliance, because I would love to enlist you in this and see if we can work together to change what right now is a very dismal paradigm. So why am I talking to you about the cancer statistics? I mean, you can go look this up online. But I want to tell you one thing, if you don't walk out of here with anything else. This disease, as you can see from this slide, is going to increase anywhere from 57 to 80%, depending on who you read, over the next 20 years. Now, that's a daunting statistic. We have never, ever had a disease where we could project 14 million new cases per year and as many deaths as you can imagine, 13 million on a global basis. Some countries, you would think, what about Africa or Asia? They're now starting to take care of some of the diseases that have taken so many of us, infectious diseases, and now cancer is becoming a global reality. I spent a lot of my time at the National Cancer Institute actually consulting with countries that are trying to put together a plan to deal with cancer. So it is going to be, I think, and actually World Health Organization just this last quarter called it the pandemic of the 21st century, and that's what it is. In the United States, it's a very daunting disease, and if you're over the age of 55, you start thinking very seriously about this disease. But we have about 1.6 million new cases in this country, and what you see in terms of the death rate hasn't changed much since the 1950s. Why is that? Well, we have more and more new cases every year, so the death rate has changed very little in these years. So we have, in any given year, we still have more than 560,000 people dying from this disease. And as I said, 77% of cancer is diagnosed in those of us over the age of 55. And the baby boomers are starting to have a huge impact on this curve. As more and more baby boomers turn 65, as you can see, the number at the bottom of that screen, if you can see from the back, sorry, it's a long way down there, are starting to turn almost daily, really increasing this burden. And the cost of this disease is staggering. It's approaching 200 to 300 billion per year, probably is at 300 billion now. We don't know really how to keep these statistics. It's an extraordinarily expensive disease. So 25% of our population will die from cancer, and 1 out of 3 of us sitting in this room will die from cancer. So, and I like to say that I don't care who you are or where you live or your station in life or how much you know, when you get a cancer diagnosis, it changes everything. And it's like a lightning bolt. And I've never known anyone, and I know just about everybody in this field, even the people in this field who know everything, when you get a cancer diagnosis, everything's off the table in terms of what you can actually do unless you have a curable tumor, which is, when we still are not curing that many cancers. And if you look at where this impact is going to be, you can see some of these diseases are really going to increase, like prostate cancer in men, and almost all men will get prostate cancer. At autopsy, almost every male has prostate cancer. We may never see it in his lifetime, but almost every male will get prostate cancer. At least 30% increase for breast cancer. Some of these are going to be staggering, though, when you start looking at kidney and some of these diseases like melanoma, which no one seems to take very seriously, again, until they get it. So these kids who, and all of us in our youth, when we laid out in the sun and soaked up all that sun, and we're now going to pay the price for it. But melanoma is a deadly disease, really a deadly disease. And once you have disseminated melanoma, it's very, very difficult to save anyone. And here's the problem. We know a lot about cancer, and I'm going to show you that we know more and more every single day, but we have not been able to actually do any better once you get metastatic disease. So once your cancer gets to be metastatic, once it moves from its origin, site of origin, into another organ, especially through the lymphatics, it's very, very difficult. And we cure very, very few, if any, of those tumors even now. And that's the reason for that is extraordinarily complex. It's an evolutionary disease, and it's one that is extraordinarily clonal. And so some of these cells are extraordinarily able and fit to be able to move from one organ to another. So you can actually take down the population of cells to nearly nothing and still die from this disease, from metastatic disease, because you have not taken out those diseases or those cells that are actually the most extraordinary in terms of their metastatic potential. So we've spent about the last 50 years thinking about cancer a little differently. We've some would say talked ourselves into believing, many of us do believe, that understanding the molecular basis of cancer is going to be the way that we actually ultimately cure this disease. Why is that so? When we sequenced the human genome, and it's been finished a few times actually, but the first time was in the early 1990s, and finally sequenced it and reported it out in 1994, it gave us a basis for understanding who you are from a molecular basis and from a genomic basis. The genes that actually make you what you are get expressed as proteins, obviously, and many of you actually know how to really find proteins better than anyone else in the world. The protein actually is the best of the class of biomarkers that we talk about today. But it's those genes that actually are actually changed or change during the course of cancer. So whether you get a mutation and you inherit it from your parents, or you get a mutation, a somatic mutation after you're born, it's those changes in the genome that produce cancer. So we say cancer is a genetic disease, which it is, but it's a great deal more than that as well. It all starts, though, with these changes, and simple mutations are not the only changes that occur in DNA, as you well know. You can have all kinds of big changes in terms of the chromosomes. You can have translocations. You can have expression of these genes that amplified, and you can have all kinds of changes that we don't necessarily call mutations. Copy number changes, all kinds of other changes. And more recently, we've found that things like microRNAs, for example, that can also downregulate genes, are very important, or proteins are very important. The proteins are very important, and more recently, the epigenome, or essentially the way that DNA gets fashioned and changed, is also very important. There are more than 3 billion base pairs that make up your DNA. And we don't have that many genes, actually, only about 25,000 genes that are active. And if you start thinking about the number of genes we've discovered that are related to cancer, there's probably around 350. Very few of those have been really called driver mutations. And I always challenge people within the genomics community to tell me what is the driver mutation. What they'll say is it's in the causative pathway for the disease. We know very, very few of those. So less than 50% of the genes we've discovered we know very little, if anything, about in terms of what the causation is relative to the change in the gene. So a lot of work to do here. There's one thing to remember about cancer, though, that cancer is tissue specific. And therein lies a lot of the, I think, direction for the future in terms of what we're going to know about cancer and what we're going to change about cancer. So when FDA, for example, if you're working on a biomarker, they say it has to be fit for purpose. You have to tell us how you're going to use it, where you're going to use it, because the seat of the cancer in this particular case is very important, or the organ is very important. So I think, I'm going to go through this with you, but I think the answer is going to be understanding the relationship between the change in the genome and the organ-specific information, and that interaction is going to be our key to understanding cancer. It's a very complex problem, but one that we're beginning to understand slightly, and I think we're just in the leading edge of understanding it. So where did we get this idea, and why do we become so excited about that understanding the changes in the genome would take you directly to understanding how to control cancer, how to diagnose it, how to cure it? It started here. A lot of work led up to this day, but this occurred in 2001. A new drug called Gleevec was discovered by Novartis in the late 1990s, or maybe mid-1990s. A very astute and smart scientist up at Oregon Health Sciences University named Brian Drucker fell in love with this drug. He thought it was really a very interesting drug, and I'll show you why in just a second. But this drug actually did something that he thought was going to be extraordinarily important. He felt as though chronic myelogenous leukemia, which essentially has been around for a long time, and when he started to treat CML patients, he discovered that they all had a similar change in a chromosome called the Philadelphia chromosome. And he discovered further that they all seem to have this change in this particular chromosome. And essentially, it's a translocation and a recombination of two parts of chromosomes to produce a BCR-able gene. We call it, it's an aberrant gene that produces an aberrant protein. And this drug actually blocks ATP in terms of phosphorylating the proteins that drive the cells that actually drive this cancer. Every single individual who had CML that he treated, it turned out, got well. These were people that were dying in hospitals in large numbers. He gave them this drug, and they walked out of the hospital. It was unheard of. It was a miracle. It's a miracle drug. And to this day, it is still a miracle drug. Not quite the miracle that when we started, but it's still a miracle drug. And this aberrant protein gave us all the impression that there just may be hundreds, maybe thousands of those kind of aberrant proteins out there. Well, we wish that were true, but it turned out not to be so true. This stands today as one of the prime examples of a targeted therapy that works, and it works for every patient that has this disease. Here's an example. It started out treating CML with this. We found out that it works for a lot of other cancers. This is GIST, or a GI tumor. And as you can see, over the course of a few months, this tumor disappeared. And this is a very effective drug for these stomach diseases as well as some other diseases. So lots of people are now treated with Gleevec. The problem is that the genome is digital information. And when you start treating it with a drug like this, what you do is you drive evolution, so these tumors become resistant fairly quickly. This is a drug, though, that since we started treating patients with it and we've understood how the evolution proceeds, what we've been able to do is re-engineer that drug several times now, so it's still very effective. I will say this is an example of what's wrong with the healthcare system. This is an extraordinarily expensive drug. It probably costs $100,000 a year to treat a patient with this drug. This is not a sustainable model. These very, very expensive drugs, these designer drugs, are not going to be the future of medicine unless we can change the way we price these things. And there's a lot of them now. So Gleevec drove in large measure this change in the early part of the last century, actually, and toward the end of the last century, toward thinking and believing that we could target changes in the genome or biomarkers, changes in the genome or molecular biomarkers, and eventually we'd be able to cure these diseases by making drugs like Gleevec for every cancer. Well, that didn't work out, and you'll see why in a second. It may eventually work out to some extent, but it has not worked out as it did for CML, where every patient, the tumor is driven by the change in that gene. It's a fusion gene. We have found one other fusion gene recently called the BRAF gene, which actually has a similar but not totally 100% effect on melanoma. So melanoma now is related to a change very much like this, a fusion gene, but it's not 100% of the patients, and the patients become much more resistant much more quickly. But the concept has actually has kind of held up. But the only way this is going to happen is if we have really, really good molecular biomarkers. And I'm here to tell you today we don't have very many of those. So if you think about what it's going to take to really achieve a vision of precision medicine, it's biomarker dependent. So you have to know the change in the gene, which translates to a change in the protein, and you have to be able to measure it, and you have to be able to measure it accurately. But essentially, this is a measurable characteristic that's used as an indicator of a biological state or a condition. I think of a biomarker as a signal that you can measure reproducibly. And if we had the kind of biomarkers we need, essentially you'd be able to inform drug development, you'd be able to detect these diseases very early, you'd be able to monitor patients on therapy, and all kinds of great, sort of future activities that our generation would love to see happen. George Post, my colleague at ASU, essentially said that, this particular area, biomarkers, represents the biggest economic benefit for personalized medicine that we're going to see in our lifetime, yet to be realized. But this is an area that is critically important to me, and I've taken it on because I think that unless we change this paradigm of how we develop biomarkers, and it's still the purview of the academic scientist is not particularly interested in rigor, and this requires a rigorous, standard-based approach. We have to change the way we do this. If we don't change the way we do it, we'll never realize that future of personalized medicine, and everybody in this room will benefit, and everybody out there that's going to get this disease and the families that are affected will benefit if we do this right. So we're all on the same page then. A biomarker essentially is a biometric measurement of biological or physical changes that's directly related to a normal or disease process or a response to therapy. I think, and believe me, this is important, it has to be clinically relevant, number one. It has to be fit for purpose, which means you have to test it in the setting that you believe that it's going to work. It has to be measurable, reproducible, quantifiable, and relate to the current state of the disease. That's all good, but unfortunately, most of that gets lost in terms of it being measurable, quantifiable, reproducible, and I'll show you why. So we actually discover a lot of biomarkers, at last count, and my friend George Post actually did this paper in Nature a few years ago, in 2013, and at last count, we had 150,000 publications claiming the discovery of a biomarker. This is a unique driver of a disease, is what people are talking about when they say they've measured a biomarker. To this point in time, we've got about 100 that are used in the clinic, and not even that many used in the clinic. It's actually probably less than 20 that are routinely used in the clinic, and in cancer, very, very few. So it's not that we don't have a lot of biomarkers. There are probably hundreds of thousands of them published, but almost none of them make it to patients. And why is that? So let me just give you a little background. And okay, I feel some culpability here. I have to admit that I am somewhat responsible for the problem that I'm now trying to solve. After the Human Genome Project was completed, Francis Collins and I sat down at lunch one day and said, why can't we find all of the genomic changes in all cancers? We can sequence the human genome now. Well, that's how this very big project got started. The Cancer Genome Atlas is actually just finishing up this year, having finished the sequencing of 20 different cancers. It was a big, big project, the largest project ever done in the history of science. And it involved something around 350 institutions. We had literally hundreds and hundreds of investigators. It was all very tightly organized and controlled. The biospecimens in the Biospecimen Core resource were all collected uniformly. They were all appropriately annotated in terms of their origin and the background from the patients, et cetera. The DNA and the RNA was extracted the same way, et cetera. So the story of the Cancer Genome Atlas was we wanted to do it in a way so that patients were actually consented in the same way, their biospecimens were collected in the same way, they were stored in the same way. We insisted on having 80% tumor nuclei in all of our samples. So this was kind of unheard of. And so we had genome characterization centers, which the National Cancer Institute supported, and all the genome sequencing was done by the National Human Genome Research Institute. All that came into a big data set that is up at the top, and when I say big, it is really the largest data set actually to date in terms of sequencing, although other areas are going to take it over. So I want to give you one slide here of what this meant. So think of this, 300 and some people, about 350 people, meeting online every single week for 8 years, and lots of meetings in between. They had to take all the changes in the genome and the proteome and actually map it to pathways that we knew were very important in cancer. And here's one example. This is glioblastoma multiforme, the most common brain tumor for adults. We haven't had a new drug for GBM in the last 50 years. Everyone dies of this disease within 6 months. We haven't changed this disease since we discovered exactly what it was and started to characterize it. But we were able to take GBM, and we were able to take all the changes that we measured in the samples, 500 samples, by the way, statistically robust sample set for every tumor, and we were able to map it to the pathways that we knew were very important in terms of that particular cancer. Now, what that's done is to bring, when we started working on this particular tumor, we probably were funding maybe 30, maybe 50 investigators to work on this cancer. There are now literally thousands of them. So data, bringing this kind of data, and on the left you see the upregulation and the downregulation of the proteins in the cell, bringing this kind of data to people brings them into the community, and when you get this kind of discovery going on, you change things. So we now have people developing new drugs for GBM based on this kind of work. The other thing I'll tell you before I leave this slide, we were able to actually subtype this tumor into 4 subtypes. And that means, and one of those subtypes actually doesn't even benefit from the drug that we were giving them. By the way, 70% of all chemotherapy does not work. We just don't know which 30% actually are going to actually work. And we don't know when we give you chemotherapy if you're getting the right drug or not, because we don't have the biomarkers to tell you that. But now at least for one class of these tumors, we know that the drug we give them, temozolomide, does not work. So this subtyping of disease, I think this is going to be personalized medicine. I think it's going to be very difficult to get down to an individual patient. If you look at every line up there, every line is a little different. So every patient's a little different. You're a little different than the person sitting next to you. If I look at your genome and you all have the same cancer, you will not have the same changes in your genome. So, but what you will have is a lot of these pathways will be in common, and I think this is as close as I think we're going to get to personalized medicine, at least in the next decade or so. So when I told you I felt responsible, it never occurred to us that when you started making this kind of data available, everyone would want to go and sequence genes, and that's exactly what happened. So now we have whole countries that are sequencing every patient. The Chinese are planning to sequence every patient, to do a whole genome sequence on the billions of patients that they have in China. The UK just commissioned 100,000 genomes. They're the first country to actually put the genomes back into the healthcare records. So we've actually started a tsunami here of sequencing, and the technology now allows people to sequence a genome for less than $1,000. So you will get your genome sequenced, I'll bet you. Because if it's less than $500, everybody can afford it. The issue is obviously, as you saw from 23andMe, it's a difficult way to practice medicine if we don't know what the changes in your genome actually mean. But by the end of 2014, there will be about 1 million genomes sequenced. That may not sound like many, but when you consider the amount of information in one genome, that's going to swamp out all the storage space we've got for this kind of bioinformatics. So about 28 petabytes by the end of 2014. We don't have computers to actually manage that kind of data. So it's very, very difficult to see how we're going to take all this information and begin to manage it and take you as a patient and figure out how to get the information we need to treat you from the genomes that we've sequenced. So this is the biggest problem we have in medicine today. It's an embarrassment of riches. We have so much information now, and we've just started. This tsunami is just starting. When you start thinking about sequencing all the patients in China, you're going to have more information than we actually have probably up to this point in terms of all the petabytes or zettabytes that are out there. So it's an explosion. And if you look at this, we're creating so much data at such a rapid rate without the algorithms and the information we need to actually analyze it. This is a problem for this generation coming up, and we need people who grew up with game theory. The kids, you know, who do nothing but go around like this all the time, they're the ones that are going to solve this problem. And if you think your kids have been wasting all their time all these years, make them go into bioinformatics. Tell them this is the secret of their success, and it will be, because I think this is the future. So biomarkers aren't doing so well. We have all that information, but if you look at the extent to which biomarkers make it into FDA for approval, these are proteins, which are the best of the biomarkers because proteins run who you are. They give you all of the actions and all the physiology that you possess. But if you look at this, we've submitted about 1,260 protein biomarkers since the early 1990s. We approve less than one per year. And it's a dismal, dismal success rate. It's fraught with failure. So as a consequence, the molecular diagnostics industry in the country is pretty much nowhere. If you want to lose a lot of money, you go into molecular diagnostics. So it's the purview of small companies right now, but there's only a couple of very large companies working in the space. And this is the reason, because there is no pathway into FDA to tell you if you have a good biomarker, and if you have a protein biomarker, this is the history. If you're working on drug development, which is totally dependent, remember, you've got to have a molecular biomarker before you can develop a drug, and you've got to have a really good one. And so if you look at the history of drug development, and this is true for cancer, it's equally true for mental illness or Alzheimer's, what have you. If you look at this, it's incredibly counterproductive to start developing a drug today. And if you look at the cost, Forbes, on my slide I have 1.6 billion as the cost, and it takes about 12 to 15 years to develop a drug. And by the way, that is the rationale for costing these very expensive drugs at the rate that they're costed at, is the companies will tell you they lose so much money here, they're trying to recover it with the drugs that they do develop that are successful. But this has to do essentially with a lack of biomarkers that tell you whether or not something is efficacious or it's toxic. So as a consequence, most of these drugs are failing in late-stage clinical trials, and in oncology, about 80% of our clinical trials are failing. So this is, again, a pretty dismal story, and one that has really driven the pharmaceutical industry to keep merging and merging and merging into only a very few companies left. And if you think that you just want to develop a biomarker, you want to do it either as a diagnostic, and it will cost a lot more money than this, or you want to do it for something else, say you want to use it actually to subclassify patients. As you can see here, the cost of developing a biomarker by the time you get it into FDA for approval, and you generally have to file a molecular biomarker as a pre-market approval, a PMA. Most of you in the room know what that is, hopefully. It's very equivalent to getting a drug approved, and it costs somewhere in the neighborhood of $100 million to develop a biomarker. So this is an extraordinarily expensive undertaking. So what's wrong, aside from the fact that biomarkers are really hard to find because the biology is very complex, but what's wrong with the field overall? The biggest single factor that's killing us in this space is the lack of reproducibility of the science. So all of those thousands of publications that I showed you have not been reproducible. So the science, and this is causing something of some fury on Capitol Hill, because people expect when you publish something that someone else should be able to do the same experiment and get the same answer, right? Wrong. So 80% of the published literature is not reproducible. And in biomarkers, it's a little worse than that. So if you look at this slide, these are family publications. This is Science and Nature. And they have written extensively on the front of both those covers was on the lack of reproducibility of biomedical science. But you know you're in trouble when you see yourself on the front page of The Economist or the New York Times or the New Yorker, all of which are actually on this, on this theme now of what is wrong with biomedical research, what is wrong with science. I should tell you that I support a lot and did support at the NCI a lot of basic science. We need basic science. We need discovery. However, we have almost no mechanism to take that basic discovery and turn it into any kind of development that's going to help a patient. And if we can't make this stuff reproducible, we're never going to get there. And it's going to be 10, 20, 50 years, and we may never get there. So this is a paper that was actually published, some work that Amgen did, trying to reproduce some significant findings that were the basis for a lot of drugs that they had spent a lot of money on. And guess what? They couldn't reproduce much of that data. 6 of the studies were reproducible. Unbelievable. So needless to say, this didn't sit so well. And if you look at it, it's probably even a little worse than that if you looked across that 150,000 publications. So I just summarized a few other things here. Do you think it's just impacting drug development? No, it's impacting across all of science and all of the areas that really should matter, whether it's epidemiology. The Amgen study, by the way, was reproduced later by Bayer, similar kind of findings. So the extent to which we can reproduce the data that is the basis for our discoveries, which is the basis for the drugs and the diagnostics we're trying to develop, is extraordinarily flawed. And fixing that is going to be our challenge, and that's what I want to talk to you about. So everybody will tell you that their data is not reproducible because this is a very complex disease. I could show you the same slide for Alzheimer's or any other disease you want to talk about. Cancer is more complex, and it's not as simple as pathways. These things are all interacting in real time in three-dimensional space, et cetera. I will give you, it is a complex disease. I give you that. And I will also give you that there's an extraordinary landscape of mutations and changes in the genes of cancer patients. I'll give you that. And the cancer itself, in different parts of the cancer, will have different mutations. I'll give you that. So it's a very complex disease. No one's going to argue with that. And that's reason to understand that you should very carefully consider when you publish something, is this real and is it reproducible? So I would say that's a reason to actually look more carefully at your data. So when I came to Arizona State University, before I left the NCI, the deputy director of FDA and I started something called the Interagency Oncology Task Force, and we decided the biggest problem we had in biomedicine was fixing this problem. How do you take something out of the laboratory in some way and have a process of development that is prescriptive and predictive, and you know that you're going to get to the end of it successfully? That's how you develop, that's how you want to develop a biomarker. So I said, let's start an alliance. This is not going to be, FDA is not going to tell you how to do this. No one's going to tell you how to do this. The communities have to do this together. And as a matter of fact, Dr. Woodcock said at one of our early meetings in a public forum that it's not FDA's job to develop the standards for biomarker development. It's the job of the affected communities. And if you look, though, in FDA's critical path document, which was published in I think 2004, 2005, essentially they're saying, she said then and she says today, biomarker development is the single biggest problem, along with clinical trials, and making better clinical trials happen is the two biggest problems we have in the whole area of biomedical science. So about 2.5 years ago, I asked a bunch of smart people to come to Phoenix and let's sit down together and figure out this problem. And I had no idea what I was getting into. And it turns out to be a much bigger problem than anyone would have guessed. And it turns out to be a more solvable problem than anyone would have guessed. So if you look at what the National Biomarker Development Alliance is, essentially it's an alliance. And our goal essentially is to collaboratively create the standards for systems-based end-to-end biomarker development. That's the only way to advance precision medicine. So what do I mean by all that? Well, if you look at what we've done, we didn't really understand any of the barriers. We've started to look at where are the barriers, and I thought I'd have one meeting and we'd have some answers. Well, we're on our 5th meeting, and we have a lot of the answers, but we still have a great deal more information to gain. So what we've done over the last 18 months, almost 2 years now, is to just identify what are all the underlying causes of biomarker failure. And you guys are going to know where most of them are. Nothing in what I'm going to say from this point is going to surprise you, I think. So if you look at the causes of biomarker failure, and I bet you in every lab sitting at this table, you could probably have told me this, but essentially it's poor or no access to really, really well annotated, high-quality biospecimens. It's essentially no control of the pre-analytic variables that go into a test. It's essentially standards, but there are no standards. There's 1,000 standards for everything, but by definition, then there are no standards. And then there are your methods and our methods and somebody else's methods are idiosyncratic, and so there are no really clear analytical methods in this space. Most of these studies are underpowered. No one oversees this process, and essentially, if you fail, well, you just fail. There's no penalty for failure. And then finally, the laboratory-developed test, which many of you are familiar with, developed in a laboratory, done by a laboratory, never seen by anybody necessarily, except the CLIA labs actually make sure your lab is functioning correctly. And recently, as you know, you've probably read the literature, the laboratory-developed test is now finally on the FDA's radar screen, It's going to be a long process, but they're starting to regulate the laboratory developed tests. So that's good news. Bad news for small companies, because that means the rigor that you're going to have to bring to these tests is the same as the one that we talked about where you were filing. It's going to cost you $100 million. So over these months, we have created a little cartoon of our own. We see this process as extraordinarily modular, starting with early discovery and late discovery. Most everything fails there. It starts right there. It's either poor specimens, wrong design, technology standards not adhered to. It all starts in discovery. And then there's assay development. Lots of issues there, especially around the availability of antibodies. The performance, once you get to that stage, you have to make sure that you could apply that to a population, and then you have to qualify your biomarker, and then you have to validate your biomarker. And if you look at it, if you don't have good starting material and you haven't really thought this through at the beginning, it's going to fail. It's always going to fail. And that's what's happened. That 80% irreproducibility is killing us, because it starts with failure at the beginning. And if you start with failure at the beginning, you're definitely going to fail. We put together a management team. It's kind of a dream team. And I've got people like Carolyn Compton, who I recruited from NCI to head up my chief medical officer, and myself, and George Post, and Ken Buto, and a lot of other good people helping us. And we have a core of probably 100 scientists that are working with us for the last 2 years. We've divided biomarkers into classes: genomics, proteomics, imaging, and complex biomarkers. Complex biomarkers are going to be mixtures of biomarkers. It could be imaging plus something else. It could be proteins plus something else, et cetera. Just checking my time, Beth, so I don't go too long. This is what we've come to. And we've decided that if you put this in some kind of a Bible verse or something, and you make everybody recite it when they start their experiment, then maybe we would get some biomarkers to succeed. So you need a relevant, biomarker-appropriate clinical question. Now, a lot of great scientists do not know how to ask a clinical question because they never see patients. And you can have a lot of really, really interesting biomarkers that are not at all clinically relevant. That's most of the biomarkers we've discovered. So if you look at the experimental design, most biologists are not statistically informed to the point where they can really design a robust experiment. Very difficult without the right input. And if you don't have the right design, if your samples are biased, if you haven't created the right size sample set, if you don't know what it's going to take to validate your biomarker, if you collect your controls 1 year and your experimental tissues another year, you automatically have bias. So there are all kinds of issues here that are causing the design to fail. Probably the most critical piece of all this is the biospecimens. I spent a lot of time when I was at NCI trying to fix this problem. People do not want to fix this problem in the laboratories because it gives them great freedom. And most of them actually love their biospecimens. They don't want to share them. It's probably a good thing they don't share them because they're not very good. The Cancer Genome Atlas, we wanted 500 samples per tumor. 500 samples. There are 350 million samples in the country. Can you guess what percentage of them passed our criteria? And the criteria, I bet, were a little bit high, but not that high. 30%. 30% of the samples in the country were good enough to use to do the kind of genome sequencing we thought was important. So that hasn't changed much. We did create best practices at the NCI, but all the cancer centers said to me, well, that's an unfunded mandate. You have to pay us to do that. We're already paying them, millions of dollars to collect samples. So I said, well, so we're already paying you, and now we have to pay you to do it right? The answer was yes. So you have to have technology standards, and there are a lot of technology standards. They're just not implemented. And the collection of the data quality is important, and having the provenance of your data is critical. And if you don't have those things, the data is probably worthless by and large, and we have a lot of data in a lot of databases that's not very good. And then finally, the analytics and the analysis. This is very sophisticated problem now. This is a very sophisticated problem. You've got to have very sophisticated algorithms to deal with this kind of data. And again, most of us are not trained in mathematics to the point that you can actually do this. So we call that our big six. But if you look at all the promise that we have, all these great technologies, all these pathways we know about, you have to start with the right stuff. You've got to start with the best biospecimens. They have to be collected in the right way. The stewardship of those specimens have to ensure that they don't degrade, et cetera, et cetera, et cetera. Things that you guys know very, very well. So does this all make a difference, or are we just, being paranoid here? We funded a lot. There's no such thing, by the way, as biospecimen science. No one's actually studied the science of biospecimens, which is, by the way, personalized medicine depends totally on the biospecimen, totally. And we spend very little money on it. No one regulates the idea. No one actually looks after a patient's biospecimen to the point that we do, say, animal rights. For example, if I give you money to hold animals, you have to show me that you're going to change the air in that room 22 times an hour, whatever it is. You can collect all kinds of biospecimens from patients, and I don't require you to do anything, except maybe get approval, from your institution to collect those specimens, but nothing else. But does it matter? Do these things matter? Yes. If you look at, for example, this is one example, or two examples actually, of the kinds of changes that we know go on all the time, like the loss of biomarker signal with time to fixation is significant here. And this is breast cancer. By the way, this caused us a lot of issues in the HER2 assay. And over here, here's a gain of signal with time to fixation. So these are all very important things. And without knowledge about the tissue processing methods and the assurance of rapid tissue fixation, protein expression data are unreliable, and I would argue that they're actually pretty worthless. So if I look at what NBDA wants to do, we want to fix this problem. What we want to do is to set up and define standards. FDA will work with us to see that those are adopted. And we'd like to do it per biomarker class. We'd like to have organizations like yours help us to educate and to provide tutorials, et cetera, for how you use these kinds of standards. Biospecimen standards, our first goal, are critical. We're holding a convergence conference in December to see if we can get these communities together, especially from the proteins and genomics communities. We want to establish a biomarker database for each step of this modular process. What kind of data do you have to have? We'll reproduce some of these results because nobody else wants to do it. And some of these critical questions, we're going to have to reproduce some of the results. And we may develop some critical biomarkers. If there aren't any for a disease, a rare disease, and we can develop them, then, and we're going to have laboratories for many of our volunteers to do this. And essentially, we're going to do this through networks and partnerships. We're not going to try to build an infrastructure. So there's an opportunity here for everybody to participate. We're trying to bring the professional societies into what we're doing so that we can get this problem solved. So I'm here, I'm a recruiter. We don't have any perks. You won't get any large checks. Maybe a t-shirt. I don't have t-shirts yet either, but essentially we would like to have. When Beth invited me, I said, yes, this is the group that can probably bring some semblance of order to what we're trying to do here, because it starts with the tissue. It starts in the OR. It starts when the pathologist gets the tissue, and it starts in your laboratory when you get the tissue. And from there, everything else is, as you know, can go very wrong. And a lot of investigators, and you all live in institutions, you know this, they don't care about the quality. They'll take whatever from the OR they can lay their hands on. But our website is up. It's www.nbdabiomarkers.org. You can go on there and find out a lot of information. And we're starting our membership campaign in 2 weeks. We got a lot of influential people on that slide, like Janet Woodcock and a lot of other people, but it's organizations like this that will help us to change this situation. So answering my question, which is what I posed, is this whole concept of molecular-based medicine really achievable in the absence of robust and clinically relevant biomarkers? No, it's not. Absolutely not. So, and what does that mean? It means that the dismal healthcare system that we currently have, where we treat disease and we treat it with no real targets. In the case of cancer, as I said, 70% of our chemotherapy is wasted. We don't know which 30% is going to actually should have that chemotherapy and which 70% should not. And the same thing is going to be true for Alzheimer's or diabetes or what have you. I believe that, and I've done a lot in this field to make sure that it advanced. I believe that personalized medicine or precision medicine or subtype or disease subtype medicine, whatever you want to call it, I think this is the future. I'm not sure, our generation will see the total benefit of it, but we're already starting to see it. It's not a panacea, but I think it will improve healthcare dramatically, and I think it has the potential of improving healthcare for everybody dramatically. And if you start looking around the globe at those numbers that I showed you, that's economically destabilizing. The countries that have healthy populations are going to be the successful populations. And I think that cancer is going to be one of those diseases, not the only one of our aging populations, but probably the most critical one. So I'm here. I'm happy to send you information about the NBDA, but we would love to get this society and your institutions helping us with this. And we will have some contract work in the future, et cetera, to give to people like you. And we hope that I hope that you will join us in this, what I think is a real tough battle, but one that I think we can win. Thank you very much.